Okay. Now the mismatch repair. How does this one work? Now, first thing you have to do is recognize which one has the methyl groups. Now, here in the E. coli system, of course, is the system that is most studied. You have this protein here. See, the mutation is right there, and this guy here is not methylated. Okay, red one is not methylated. The green one is methylated. So there is this protein here called MutS that is going to check out for problems in the DNA, and then if it detects, it is going to call into this MutL for fixing the problem. So how does that work? Okay, so let me just show you here, because it's explaining it very nicely.
Although rare, incorporation of base pair mismatches during DNA replication does occur. There are DNA repair mechanisms that can fix such mistakes. One such mechanism is the methyl-directed mismatch repair system. In Escherichia coli, this repair system can detect a mismatch shortly after DNA replication and repair the newly synthesized strand, thus preserving the base pairing that was present in the original DNA molecule. The system relies on the fact that methylation of a newly synthesized DNA strand does not occur until several minutes after the strand is made. This provides a window of time during which the newly synthesized daughter strand in a DNA double helix can be distinguished from the parental strand, which is already methylated. A sequence of DNA with only one methylated strand is said to be hemimethylated. Actual recognition of a base pair mismatch is the role of the MutS protein. MutS binds to a mismatch site and forms a complex with another protein, MutL. The formation of this complex recruits MutH to the region. MutH binds to a hemimethylated site. The MutS-MutL complex interacts with MutH by a DNA looping mechanism. MutH then makes a cut specifically in the non-methylated strand. An exonuclease digests the non-methylated strand from the cleavage site to just beyond the mismatch site. This leaves a gap in the daughter strand, which is filled by DNA polymerase III and then sealed by DNA ligase. The end result is that the mismatch has been replaced with the correct base pair as indicated by the original DNA strand.
So now you know exactly how the system works. You have the MutS that will detect the mutation, recruit the MutL, and they will know which side of the DNA is the one that is methylated. After that, you also have more details here. If you remember, just MutS and MutL and the MutH and the complex it forms here. Now, since you're going to see several forms of DNA repair, how do you know which one is which? Okay, this type, let's say mismatch repair, this one is the nucleotide excision repair, right? So here, look, if you think about it, MutS, how is it that the S looks like? It has curves. Okay, you have a curved shape S. So now you know that this process is going to be curved. So you have proteins that are going to interact. How? It's going to know, it's going to detect which one has a methyl group, and it's going to have to bind on the other side in order to get the parental strands on one spot and recognize the ones that are newly synthesized and then degrade that piece that has the problem. Now, this type of excision repair, it does not only take that base out, that is the problematic one, it takes a whole chunk of DNA, close, after the methylation site and close, passing the base that has the problem. Okay, so after it degrades the newly synthesized strand, then you have DNA polymerase that is going to get in there and reconstituting. Okay, so most of the repairs, as I mentioned, require one good strand that is non-mutated. Now, when you have a problem that you have a cut on both of the strands of the DNA, how is it that the cell is going to know how to repair this? Maybe there are some bases that got cut here in the middle, or maybe you have a piece of DNA that got added over there, and you don't really know how is the frame running. You don't know if you have a frame shift or not. And if you repair something with a frame shift, you're most likely going to have a problem for the expression of the protein. So there is this other form of DNA repair that is called a homologous recombination repair, and basically uses the same type of mechanism as you would form with recombination. So what happens here is that once the cell detects that it has a double cut, you have some enzymes, the REC-BCD, they'll recognize the double strand cut. Now, why is it REC for recombination? Okay, REC-BCD for recombination. That's the recombination mechanism. Now, once this enzyme will recognize the double cut, it's going to make overhanging sites on the problematic DNA here, on the one that has a double cut. And what it's going to do is going to recruit the homologous pair for this chromosome, because it's going to use the old non-mutated homologous pair to fix up the one that got the double cut. So it's going to pair up with the non-mutated chromosome, and now these pieces here, you see, these pieces here, they are overhanging over there. They're going to invade the new DNA, the DNA that just got there. It will invade the DNA, and you'll just copy a piece of the good DNA and then repair itself like that. It's just like recombination. It's like, oh, I have a problem, I have a double strand break here, just bring me my sister, I need big help. So you get the identical sister here, oh, just give me a piece of your arm, I'll give you the one that is, broken in half, so just repair it, just like it happens in real life. Right? So yeah, sibling support. Although I do sometimes feel like breaking the sibling arm. Or anyway, yes. So this is one type of recombination repair that is used when you have a double cut. So remember, you have to invade the good loop, the good DNA. Okay. Now there is another form of double cut repair, which is a more crude form of repairing this. I would call this the desperation double cut repair, because when you don't have the sister chromatid readily available, what do? You dare not just leave this DNA floating around. You're going to lose the information. But once you cut a piece of the DNA, double cut in there, you know what happens to DNA that is cut, right, in the cell? It's going to get degraded. It's going to be chewed up. And then you're going to lose a piece of the arm. Okay, you're going to lose a piece of the DNA. So in order for that not to happen, the cell has this other form of non-homologous end joining repair. Okay, so what does that mean? That means that the homologous chromosomes are not involved at all. This one is a, oh, just fix yourself type of a repair. So here is a poor guy with the cut arm, and there's no sister. He's the only sibling in the household, and he has to fix itself somehow. So notice that before it can be fixed, first thing, it has to be stabilized. Because as I mentioned, if the DNA is floating around the cell and is a piece of DNA that is not protected, it's going to be chewed up. So first thing you do, you get stabilizing proteins there. Then you're going to get end joining proteins attached, and you're going to just join the ends back together, just like a gluing back mechanism. Now, the advantage of this system is that it can repair any type of double strand cut in the cell. It just glues back together, so you don't lose a piece of the DNA. The disadvantage of this system is that if you had lost some information in between this cut, this double strand cut, then you wouldn't know the information is lost. So you're still gonna get frame shift, and you're still gonna get some type of mutation. But then the cell is thinking, I better just have frame shift in one gene and not lose all the rest of the genes that are in the rest of the chromosome. It's a matter of survival. Okay, so the only thing here, it's noticed that I did not mention specific proteins here. So what is important to know here is that you have end binding proteins that stabilize the DNA. And then you have this cross bridge protein, these blue ones here. And what is it that they're doing? first you're stabilizing it, and then you are cross-bridging it. You want to get it glued back together. And then once you have it cross-bridged with these blue ones, you just get the ligase and paste it back. That's like a blunt end cut that got snapped back together. And you can imagine this is also the source, right, as I mentioned, of DNA mutation. When you have two chromosomes that are close together, when they're structured, two chromosomes close together, and if it happens that both of them have a cut on the double strands, if they are very close together, they can just swap those pieces. Then you have what type of interaction there? Reciprocal, non-reciprocal. You have a translocation, reciprocal translocation. So they can just swap the pieces, right? Now, the last type of repair mechanism is a type of SOS repair mechanism for the cell. This mainly happens in E. coli, and emergency proteins, they get upregulated, and the cell learns how to deal with the DNA damage, and it just moves on. It's a type of survival mechanism. I'm not going to go into details of this. Okay, so that is as much as you have to know for DNA mutation and repair.